So we're going to move from the entire mouse in the next two talks down to these wonderful new things called organoids. And Parul is an assistant professor at University of Texas, San Antonio, and she's going to tell you about this magical new system. So Parul, thanks very much. Actually, I'll hold on to this.
Hello, everybody. Can you hear me well? Okay. Thank you so much to the organizers for having me here. It's really an honor to be present here and present the little work that we have been able to do for past 6 months, actually. But we have been in touch with the foundation for 2.5 years, and it has taken us some time to gather resources and do everything that we can talk about today. And which one? Oh, okay. So before I move forward, this is the team, 3 of us. All 3 of us are present here. Dr. Zane Librand is an assistant professor at the Department of Biology at Texas Women's University, and Preetika Karamcharya is actually his graduate student. And none of this work that we are talking about or we are passionate about would have been possible without their collaboration. And it is actually Preetika's hard work that we have been able to generate some neurons and get some preliminary data that I can talk about today. So brain is, and thank you, first of all, like all the speakers before me, and Sharon, I don't think I can match your presentation at all. And when I come to my Western, please look away, okay? Yeah. So thank you for such great introduction on all the basics of the techniques that we use in the lab. I don't have to do that. And Dr. Zeynep Turmer, I have used a lot of your modeling of the protein in my slides, as you will see, because I love those figures. So brain is one of the most complex organs. There are 100 billion or 86 billion neurons in our brain, and each neuron forms connections close to approximately like 1 quadrillion, which is 1,000 trillion connections. So just a brief recap again, this is how a neuron looks like. And you can see, if I can, is there a laser pointer? Oh, perfect, yes. Oh, yeah, it goes far away. Okay, never mind. That's okay. So, and this is where, like, you see the synapses are actually formed between the axon terminals and the dendrites. And the zoom-in version is the synapse, the actual synapse where we are all talking about the PSD95. It's present on the synapses. This is the place where it is present. So as we learned this morning in the very first lecture from Alex, that this is a DLG4 gene is present on chromosome 17. It has 22 exons, and we all got a brief introduction on how the exon splicing is done, how the mutations can affect these, and it is approximately 27 or 30 kb gene. It encodes for the protein PSD95, which is a postsynaptic density protein. One of the things that I really want to emphasize is this protein is really beautiful in the sense that it has very beautiful modular domains, which is very critical for its function. And I will walk you through those functions a little bit in the next few slides, that why each domain is important and how a mutation in any of these domains can have such a pathological consequence, right? So, and again, this is just like these are the places where the synapses where this protein will be present. So if you look at the structure of this protein, there are 3 PDZ domains. There is a Sark homology domain, and there is a guanylate kinase domain. So this is from Dr. Turner's lab publication. Here you can see that the PDZ1 domain, it actually, we have been learning that it binds to receptors, but this, I'm trying to emphasize that how the N-terminal domains binds and how the C-terminal domains bind, and how all of these are so critical because it is like a glue that's holding everything together. And if anything happens to any of these structures, there can be pathological consequences. So you see the PDZ1 domain binds to the NMDAR receptors, which are very important. These are the glutamatergic receptors in these synapses. Then they also bind to the AMPA receptors. You can see that PDZ domain, 2 domain binds to the potassium channels, which are the shaker-type voltage-gated potassium channels. Then the Sark homology domain and the PDZ3 domains, first of all, they bind to the other receptors on the membranes. And they also bind to SYNGAP, the SAPAPs, and the SHANK, and the actin. All of these C-terminal domains, like Sark homology domain and the guanylate kinase domains, these domains are important for the structural organization of the actins and the other proteins intracellularly, and the N-terminal domains are important important for the organization at the receptor. So if anything happens to any of these domains, the structure can fall apart and can have pathological consequences. So how do we study this in patients? Like Sharon just talked about in mouse, we are trying to study these in patient cells. So what we do to study these is we take these patients or healthy subjects, use their blood to convert them into something called as induced pluripotent stem cells. Now these pluripotent stem cells are the cells which have the potential to generate into any cell type of the body. And we are particularly focusing on the neurons. You can either grow them as 2D neurons, or you can grow them as a 3D brain organoid structures. The next speaker will talk about that, and I will focus only on the 2D neuronal structures. So the advantage of using these model systems is because they recapitulate what patient has, and they can also be used as a good therapeutic screening because you can do it in a high throughput fashion. You can grow these neurons in multiple wells in a plate. You can do drug screening. You can do other sorts of screening in them. So this is a very robust model system, and we are trying to really optimize it in such a way that we can help the foundation, we can help in understanding the phenotype, and also bringing the therapeutics. So the big aim of the project, what we do, how do we study this? Again, as Sharon described, we look at the expression of PSD95, which is through the Western blot and through the immunohistochemistry. The immunohistochemistry can also tell us where exactly in the neurons, in the synapses, this PSD95 is present. Is there a difference in the number of neurons in these patients? Is the localization different? The dendrites are affected. And the Western will tell you the overall abundance of the protein, of the PSD95 expression, if it is upregulated or if it is downregulated in the patients. And then the next aim of this project that we're trying to do is have the electrophysiology, like to understand the electrical signals of these neurons, and that we do through something called as a multi-electrode array. And that's what Dr. Libran's lab specializes in, and that's why we are collaborating on this. So you can plate the neurons in these multi-electrode array wells. They can record the activity from the neurons, and then you can find out whether the activity of the patient neurons is different from the activity of the control neurons, and kind of finding a biomarker for any future therapeutic screening. So with the generous support of DLG4 Shine Foundation, we have 5 patient iPSC lines. I don't think we would have been able to do anything if DLG4 Shine Foundation had not helped us with this. We have these 5 mutations, and instead of just reading through this, let me show you where these mutations are in the protein. And also, like you can see, like we have a representation of mutations across the protein. So the very first mutation we have is actually in the PDZ2 domain, which is the 43P8, which is the female over here. Then we have a mutation in an uncharacterized region between the PDZ2 and PDZ3 domains. We also have a mutation AR79, which is in PDZ3 domain. And then we have a mutation in sarcomology 3 domain. Now, looking at all of these, we also have a splice variant mutation, which I've not mapped onto the protein. But we have total 5 mutations. And you can see, like, each mutation, we think that it is a protein truncation mutation, except the VCFQ, because that is hopefully the full-length protein, and it is not degraded. We still have to check that. But most of the mutations, the 43P8, YTQ8, and AR79, they are probably protein truncation mutations. And we are still figuring out whether they hang around or they are actually degraded through the NMD pathway. But you can imagine when I showed you in the first few slides that all of these domains are really important for bringing together the synaptocomplex of the synapses, the postsynaptic density compartment. Like if any of the mutations are affecting these domains, then that complex can fall apart really easily. So what we have really tried to do in the very first of our studies is like use AlphaFold server. I don't know if you're familiar with that, but in the past, people would do a lot of X-ray crystallography to understand the structure of the protein. Nowadays, through the help of AI and through the computational advancements, you can look at the structure of the protein. And we have tried to understand if these mutant proteins may have a different structure. We are not going into details of which amino acid is present where and how they are changing the overall structure. But what I'm going to show you, and I hope you can appreciate, is that this is how a control or a wild DLG4, wild-type DLG4 looks like, the PSD95 protein. The very first mutation that I'm talking about, the 43P8, which is the mutation that actually is the full length, but with the mutation, that looks similar, but it may be completely misfolded. It may bind to something else that it should not be binding, and that could be the cause of the pathology. Then you have YTQ8, you have AR79, and you have VFCQ. All these three proteins these mutations are protein truncating mutations, which means that maybe these proteins are degraded, but if they're hanging around and not degraded, then this is how misfolded or their conformation can change. And if their conformation changes, everything downstream of them can be really affected. So this is just a very crude modeling of these mutations that we are trying to understand about these proteins. Then next, what I'm going to talk about is after this clues from the AlphaFold, we are trying to actually generate neurons from these patient neurons, patient iPSC lines and the control iPSC lines. And this is just a brief protocol that we follow. We take these iPS lines, we dissociate them into single cells, plate them onto the cells, and then we grow them for almost 21 days, which is 3 weeks, and try to push these iPS cells into the neuroectodermal lineage, and then give them some more factors so that they differentiate into neurons. So around 21 days, we start putting them into the growth factors that can help these iPS, which has gone into the progenitor phase, so that they can differentiate into the neuronal phase. Basically trying to grow neurons in a dish out of the patient cells. So this is the very brand new, straight from the oven data, which we got on Tuesday, and we all came in on Wednesday. So this, you're looking at neurons in a dish. This is from the Librand lab. And you're looking at, we have been able to do this with only one control and one patient line so far. And we have a lot more controls to add. We have a lot more QC steps to add. We have to repeat a lot of things. But here, I hope you can appreciate that we have been able to grow neurons in a dish. And all the green that you're looking at is the neuron, which has been labeled with a marker called TUJ1. It is a cytoskeletal protein that you can stain for. And then all the red dots, little tiny red dots you're looking at, if you can see here. Okay, so it does not work. Those are the PSD95 proteins in these neurons. So I'm not concluding anything as of now because we have to do a lot of quantification with the correct QCs and the correct controls. But what we can see is maybe there is less PSD95 in AR79 patient lines. We will do dendrite quantification. We will look at all of those things later. But this is our data. And then next, please, Sharon, please look away. This is the Western that we tried to do. GapDH didn't work, which is strange, but and a lot of other hiccups. This is, again, I did this, finished this on Tuesday, put it in the slide, and flew yesterday. So I didn't even have time to go back and repeat any of the steps. But I promise I will have better Westerns and stainings next time. So what we are, again, trying to look in the patient neurons, is there a deficiency of PSD95? Is this truly a haploinsufficiency or not? And we are going to do this by Western. We are going to do this by immunohistochemistry. And I think in humans, PSD95 is around 80 kD, but I'll have to double check that. That's what my understanding was. But so far, what I can see, and I did try to quantify this, it looked like AR79 does have less PSD95, but again, do not quote me on that till I have done my proper controls and everything. The last thing that I want to talk about is the aim that I talked, we are looking at the activity of these neurons. So this is what Librand Lab is also doing. They are using the MEA, multi-electrode array system. They are looking at the electrical signals from the control and the patient neurons. What you're looking over here at is all the ticks are basically a spike, an activity from the neuron. And right now, what we are seeing, these are just 14 days old neurons. So 21 days in the SMAD inhibition, where they're growing into, where they're becoming something that can give rise to neurons. Then they are a week for becoming neurons. And then for 14 days, they are neurons. And that's it. They're very young neurons right now. They're not mature enough, probably. But we are seeing some activity in both the control and in the patient neurons. But right now, we cannot say anything if there is a difference in the properties of the neurons in the patient and in the controls. We hope that there will be, we think that there will be a difference in the activity when they mature more, which is around like 60 days, which is 2 months. So these are just 2 more graphs, which are just showing that we don't see a huge change between the patient neurons and the control neurons so far. But our guess is, and if we are thinking it right, there will be a difference as these grow more, actually. So with this, the big take-home message is what we are trying to do is we are taking the patient cells, blood, we are converting them into iPSCs. We are differentiating them into a 2D neuronal culture, basically growing neurons in the dish. And we are trying to understand the phenotype. We are trying to find a biomarker that would be the most reliable biomarker to proceed with any therapeutic screening that we want. So that is the stage where we are at right now, understanding the phenotype in the dish. Does this phenotype actually resemble, just like Sharon said, in like mouse, does it actually resemble what's happening in a patient? Can we be close enough in the dish to say, yes, this is what happens in the patient, and this is what is happening in the neurons in the dish as well? And I think that would be the gold standard to define when we are working with a project like this. And once we have achieved that, then definitely we can move into the next stages of like finding the compounds that can actually help in the increased expression of PSD95, or if there is more, how to decrease the expression of PSD95 and all sorts of things. But right now, we are just at this stage. We're trying to figure out how these new patient neurons are different from control neurons in terms of PSD95 expression, in terms of electrical signals, in terms of these things. We are also trying to understand, we have written some proposals, like how the binding partners of these mutant proteins are affected, but we'll see how that goes. So acknowledgements, Dr. Zainar Librand, he's my friend, he's my collaborator in science. I mean, I go to him with like, I can come up with an idea, and I go to him, and I'm like, Zaina, I have an idea. And he's like, oh, dear, again, OK. So but he's always on board, and none of this would have been possible without him coming on board, without his lab coming on board. Preetika, she is the one who has been working on these iPS cells, who has been growing these neurons. My supervisor, Dr. Jenny Shea, whom I work with at UTSA, Brain Health Consortium, I'm part of that at UTSA, and of course, patients and families, and DLG4 Shine Foundation for trusting us and for giving us this opportunity to be part of this community and to be able to do something that we think like we're living right, actually. And funding, we wrote so many grants, and finally TWU came to our rescue. This work has been possible by a very small funding, a seed grant from Texas Women's University. So we are very thankful for that. Thank you.